to have Dr. Frank Chuang talk to you guys about biophotonics and cancer medicine. Okay, should be a very interesting topic and relevant to what some of you guys are doing for your term.
Okay, so hello again. Good to see you all since Monday when we had the tour of the CBST lab. So my name is Frank Chuang, and my role here is to give you a presentation about how biophotonics is actually being used in cancer medicine. Sorry. I can give on a couple inches of my leg. All right. So when I talked to some of you on Monday, I asked how many of you were pre-med, and actually it looked like a third, maybe even half of you are all pre-med. Is that right? So, yeah, no, maybe. All right. Hopefully I don't scare you away from this. I think it's a good idea. Definitely that you're taking this class. Being in medicine these days is totally different from being in medicine, say, 10 years ago. More and more, there's talk about interdisciplinary science and interdisciplinary research. It's not enough just to know physics or chemistry or biology. You really need to be able to integrate all these different sciences all at the same time. And one thing that we hear a lot of these days is something called translational research. And this process, this process of translating science into medicine requires basically basic scientists who make the discoveries about, laws of nature, how the body works, mechanisms of disease, and they take this knowledge and eventually somehow they have to get it into medical practice. Okay, but the way to do it typically is through the development of biotechnology, and this is the domain of engineers. Okay, so it's the engineers who will take some new discovery about science, come up with some interesting or cool gadget that can be utilized in medicine to either perform advanced diagnostics or perform some kind of new therapy. And in the context of cancer, all of these elements are important. So the goals of this presentation, got 3 of them, basically to review some basic processes in cell biology and introduce some key concepts in cancer biology. All right. Also to describe the primary tasks in cancer medicine. These are, number 1, identification. Number 2 is localization. Number 3 is treatment. And then 4, kind of our ultimate goal is prevention of disease. All right. And then finally is to demonstrate how biophotonic tools and techniques are actually being developed to advance cancer medicine at all these different levels. All right. So I missed the first part of Anna's talk, but I know that she gave you an excellent introduction to molecular biology, and hopefully her talk has helped pave the way for me to talk about cell biology. As you know, we have a lot of different molecules that make up our bodies, and these molecules are organized or grouped into proteins, nucleic acids, carbohydrates, and lipids. All of these have single units called monomers, which if you put together in large strings, you'll get certain large molecules called polypeptides. You get DNA, you get polysaccharides, you get triglycerides, right? And so this is kind of a breakdown in our body of the dry mass. All right, we're approximately 50% protein, 18% nucleic acids, 15% carbohydrates, and 10% lipids, right? Don't forget that this is what I say dry mass. What I'm saying is that that's 20% of our weight, right? The remaining 80% is water. Okay. I don't need to go over this, but basically gene expression is the process whereby DNA in the nucleus contains all the information, basically the blueprint for creating the whole organism. The process of transcription and translation allows for the DNA to create all those other building blocks, the other molecules that we were talking about. All right. Did you go over this? Any? Okay. All right. So I won't dwell too long on this, but basically to review eukaryotic cell structure. All right. Just remember that a cell is a basic unit of a living organism. Nearly every function that's in the living body can be represented at the cellular level. All right. Cells need to breathe just like we need to breathe. They need energy and nutrition just like we do. They're involved in communication. I'm going to be talking a lot about this, both inside and out of the cell as well as within the cell itself. Okay. So the human body has organs and in the same way the cells are made up of different organelles. Important organelles to realize we've got the nucleus that contains genetic material, mitochondria, which are battery pack, or kind of power plants. Endoplasmic reticulum are the factories where proteins are manufactured and packaged for transport. Plasma membrane is the skin, right, that separates the cell from the outside environment. You have microfilaments, microtubules, which form the skeleton of the cell. Okay. You put all of these things together, and one of the main activities that a cell is responsible for is dividing and multiplying, right? And this is called the cell cycle. Now, this is kind of a looping picture of a cell that's undergoing mitosis, right? And most of you, I'm assuming, have studied this process before. And probably you know about this cell cycle as being a very tightly controlled, well-orchestrated process where the different organelles are reproducing and then subsequently divided before the whole cell will split into two daughter cells. Right? There's a really terrific online activity that you can, if you go to the Nobel Prize website, there's an online activity that you can go to where basically it coaches you through the process, basically you being the director of a cell to direct mitosis, and then you'll come to appreciate all the individual steps and checkpoints that are necessary in order for you to have a successful mitotic division. Right? Once you've appreciated that, you'll appreciate that cell signaling becomes an important process that is required. And so cell signaling is the process of combining both internal and external communication in order to carry out many complex activities. All right. Cell signals can be carried out either through chemical processes or electrical processes. An example of chemical would be hormones in the body, right? So hormones travel from your brain to other parts of your body, and it's those molecules that will signal to the cell to do something, right? When I talk about electrical signals, mainly I'm talking about the nervous system. All right, nerves will conduct electrical pulses throughout the length of the fiber and then transmit signals going from your eye to your brain. The overall activity of a cell is determined by kind of the sum or the integral of all the individual signals that it's receiving. And so you can imagine the cell being a very large machine that's just getting messages from all different directions. And what the cell decides to do depends on kind of the overall aggregate of all the individual contributions of the signals. So some signals are telling it to go, other ones are telling it to stop. What the cell does in the end is kind of the average of what all these different signals are telling it to do. Right. So when we talked about extracellular communication, we're talking about signaling that occurs through the cell membrane, right? So the cell membrane contains many different receptors as well as ion channels that enable it to communicate with the outside world. Examples of receptors include what are called Gproteincoupled receptors, and these are receptors that'll recognize a lot of important hormones, one of them being adrenaline or epinephrine, glucagon, which is involved in the utilization of sugar in our body, and serotonin, which is another neurotransmitter. We have also receptors that have enzymatic activity on the inside, and I'll talk a little bit more about this, but these are receptors which will control cell growth, for example. So this is going to become very important in cancer. There are also ion channels, and this is especially important in neurotransmitter function and also nerve depolarization, but these open the cell membrane to allow ions to pass through, so it affects the charge that you'll see across the cell membrane. Right. So Gprotein-coupled receptors, quickly. This is the Gprotein-coupled receptor up here. It's characterized as a long protein that has 7 transmembrane domains, so it kind of snakes in and out of the cell membrane. It's a complex structure. But it's well known because it's named as Gprotein-coupled receptor because there's this molecule that sits on the inside of the cell membrane, all right, and it's called the G protein. Now, the G protein couples to an enzyme which is also attached to the cell membrane, and this basically converts AMP to cyclic AMP. All right, and cyclic AMP is an example of a second messenger, which then can travel into the cell towards the nucleus or to other parts of the cell in order to cause an effect inside the cell. So this process allows for a signal on the outside to cross through the membrane and get transmitted into the rest of the cell. Right. So these Gproteincoupled receptors are involved in the perception of light. They allow you to smell. They are also important for mediating the signals transmitted by hormones and neurotransmitters. Right. The receptor tyrosine kinase pathway is another example of a membrane protein, but instead of using a G protein to transmit the signal into the cell, it actually relies on like a relay race. It uses phosphate groups as kind of the flag that gets passed from molecule to molecule until it ultimately is carried into the ultimate target inside the cell. And so you'll have a receptor on the outside, for example, the insulin receptor. Okay, so insulin on the outside of the cell gets bound by the insulin receptor, and when that happens, it triggers phosphorylation to occur on the inside of that receptor. That phosphorylation is recognized by molecules inside the cell and will cause what's called a phosphorylation cascade. All right, basically, it's kind of an avalanche of phosphorylation activity where the phosphate group, this phosphate is the same phosphate which is in ATP. Right, ATP is adenosine triphosphate, so it has 3 phosphate groups inside. Right, and so one of those phosphates gets transferred and attached onto some signaling molecule, and that signaling molecule then goes from being inactive to active. Right. But basically, this is another important process, and this receptor tyrosine kinase, an example is the endothelial growth factor receptor, and so this is EGFR, and this stimulates skin cells to grow and proliferate. And EGFR is actually going to turn out to be an important receptor when it comes to looking at cancer. You might ask the question why signaling is so complex in these things. Why is it that it's necessary to have so many different molecules involved? There are 2 reasons. One of them is signal amplification and propagation. Right? How do you get a single molecule of a hormone to cause an effect on a whole cell? Right? The only way to do this is to have that small hormone get amplified through kind of a chain of events. So if you have an enzyme, right, the enzyme gets turned on and can affect 100 or 1,000 different substrate molecules, and those substrate molecules will also have an effect on 100 or 1,000 other molecules. So you can see how the effect of one single molecule binding on a receptor will have a very large effect on the inside of the cell because you're recruiting more and more molecules in the process. This also gives you a chance to kind of feed in some side signals and have some kind of variation in the response. So my response to adrenaline as opposed to caffeine might be different. They both are stimulants, right? But then the actual effect physiologically might be subtly different because one stimulant is coming into the pathway differently from where another stimulant is coming in. Okay. So, like I say down here, another reason for the complex signaling behavior is that you want the opportunity to be able to modulate that signal. Right? Sometimes you want to turn on the mitochondria, but you don't want to turn on the nucleus, or you want to turn on some other organelle, but not some other system. Right. So once you understand all that, and we understand about cell cycle, cell division, then we start getting into more complex issues. All right. For example, how does an egg, right, which starts out as a single fertilized cell, turn into a whole organism? All right. You turn into a human being that has hair, 2 eyes, 2 legs. You have a heart. You have liver. You have intestine. All these different systems come out of starts out with the division of one cell, right, into 2, 2 into 4, 4 into 8, 8 into 16, and so forth. So at some point, these cells have to start to differentiate. Okay, this cell knows that it's going to become a head, one cell inside the head. This one knows that it's going to become a cell inside the foot. And so this whole process is called developmental biology. Okay, and so there's a whole science associated with this. But basically, it's the process of how does the cell know up from down, left to right, And in the beginning, so this movie is showing you basically what starts out as a fertilized egg, all right, starting to divide. Now, this is the yolk sac. So this doesn't start out as a cell, right? This is actually turning now into the embryo. But basically, this is not a human, obviously, because humans don't have yolk sacs. We have an umbilical cord and a placenta. So this is probably the embryo of a chicken, right? But it starts out as a glob of cells that are attached to yolk sac. The yolk sac simply provides the energy and nutrition for the embryo to start growing. But you can see how it starts from just this homogeneous, nondescript clump of cells into something that definitely looks like a head. You start seeing an eye forming here, what looks like a spinal cord and a tail. Right? Cool. So clearly, in the beginning, the cells are undifferentiated. They're called pluripotential because there's a number of different fates that cell could have. But what are the factors that determine which way it goes becomes a big question. So clearly, elaborate signals, both internal and external, guide the proper development of the embryo. Right, and this is an example showing you how you go from one of these nondescript stem cells, okay, into all the different types of blood cells that you have inside your body. And so inside the bone marrow, you have what are called hematopoietic stem cells. These are not like true embryonic stem cells, which I'll talk to in a little bit, but these are called adult stem cells. All right, so these are stem cells that could form any particular type of tissue, but they can't cross over. So it could be a neural stem cell, it could be a muscle stem cell, it could be a hematopoietic stem cell, which means that it could become any cell within that particular class of tissue, okay, but not cross over into a different type. And so in this case, hematopoietic stem cell is supposed to turn into either a red cell, or it can turn to any number of these white cells, or even lymphocytes, right? And so this describes the process. Normally what happens when a cell divides is that every time a cell divides, it moves down one of these paths. All right, so the daughter cells are never quite identical to the parent cell. All right, the parent cell is a little less differentiated than the daughter cells. The daughter cells are always moving in the direction of becoming a fully differentiated cell. All right, only stem cells have the capability of regenerating themselves into two daughter cells that are exactly identical to the parent stem cell. And that's what makes stem cells so unique. Right? So that's kind of what I'm saying here, is that in terms of understanding stem cells, adult stem cells versus embryonic stem cells. All right, adult stem cells basically they can renew themselves continuously. All right, and depending on the need, if you injure yourself, it senses that it needs to produce more daughter cells to fill in this particular area. It'll start to do so, but otherwise, in normal housekeeping maintenance mode, it'll simply divide and create more daughter cells like itself. Okay. In contrast, embryonic stem cells can become literally anything. Okay, because these are the cells which you originally start out with as a fertilized egg. Okay. This diagram is simply showing some of the research which is going on in trying to understand some of the chemical factors that kind of instruct a stem cell to start differentiating and to try to figure out, well, in this case, what's necessary to turn this embryonic stem cell into a neuron versus turning it into a pancreatic cell. So life and death of a cell, all I'm trying to say here is that normally differentiated cells do not divide indefinitely. Okay, there are these structures in the chromosomes that are called telomeres, and this is kind of this, what happens is that did you talk about this? Okay, so in the process of DNA replication, all right, you have a machinery that's responsible for reproducing a strand of DNA, and that machinery basically needs pavement in order to run along the track. And so when it reaches the end, if it can't reproduce the very, very end of a chromosome unless it has a little extra space to move, all right, and that extra space is the telomere. Okay, so the telomere gives it room to reproduce the DNA all the way up to the very end of the important coding regions. All right, and what happens is that the telomere gets shorter and shorter after each replication process. All right, so a cell can only undergo mitosis a certain number of times before it stops dividing again, because there's simply no way that it can reproduce the chromosome all the way up to the very end. All right, So this is kind of a new area of research, but people are trying to understand, well, maybe if I lengthen the telomere, I can get the cells to reproduce for a longer period of time. So people are kind of trying to find the secret to eternal life by being able to get these cells to live forever. Okay. But there's another process. So basically, when the telomere runs out, right, that's when that cell dies. Okay, and if all the telomeres in your body runs out, that's when you die. So that's the normal case. And then in certain instances where there's disease, there's another process called apoptosis. All right, and this is where a cell recognizes that there's something wrong with it, and it basically sends the instruction to kill itself. All right, so it's kind of a cellular suicide. And this is also a very, very interesting process that researchers are trying to better understand. And this is a picture of three cells, I think. So this one's a normal cell, whereas these two have undergone apoptosis. All right, and so they basically kind of destroyed themselves, and you can see that they've lost all sort of form and function. So in terms of understanding the whole machinery of life, we've got a number of new different sciences. Genomics you probably have heard about, which is the study of gene structure and function. Proteomics is kind of the step past genomics, which is the study of protein structure and function. Right? And if you hang around the people that I hang around with, you'll hear all these phrases like metabolomics, which is the study of metabolic pathways and, you know, what are the components that make it up and what's their function. So people are basically going from single molecules, going to cells, and moving up and up in kind of the biological hierarchy to kind of put together how the whole machinery of life can be determined from just basic molecular mechanisms. Right. My question to you is whether, you know, can you really explain all of life just by molecular mechanisms, or is there something else going on? That's more of a philosophical question. Okay, so now finally we get into cancer. Okay, what I've tried to do really quickly is explain to you what happens normally in a cell. Now let's start talking about what happens abnormally in a cell. All right. So some of the terms that you'll often hear when you hear about cancer is tumor. Tumor is basically, or it's also called the neoplasm. Neoplasm meaning new growth. All right, is any abnormal growth of cells or tissue. All right, tumors can either be benign, which are considered non-cancerous, or malignant, which are cancerous. Right. Cancers are malignant tumors which grow rapidly and they can metastasize or spread to other parts of the body. All right. There's more than 100 different and distinctive cancers. These are not contagious. All right. They will spread inside the body, but you can't give cancer to somebody else by touching them. Although there is something else I'll mention something about infectious disease and cancer a little later. All right. More basic cancer facts. Cancer can involve any tissue of the body, and they're named for the type of cell or organ in which they start. And so if a cancer starts in your lung, that's lung cancer. If you have a cancer in your brain, that's brain cancer. If you have a tumor in your brain, but if you look under the microscope, it looks like lung cells, it's probably a lung cancer that metastasized to the brain. So it's not called brain cancer, it's called lung cancer, which is metastasized. All right, does that make sense? So that's what I'm saying. Even if cancer spreads to another part of the body, it's still named after the tissue in which the primary tumor started. Right. Skin cancer is the most common type of malignancy, followed by prostate cancer in men and breast cancer in women. Actually, that may not be. I'll check. Lung cancer keeps on climbing. I may have a separate slide here that says that lung cancer is actually now number one. Like I said, almost any kind of cell can grow into a tumor. All right, it doesn't have to be cancer. So there's something called a teratoma, and this was something in medical school that everybody got really grossed out and fascinated with. But basically what these are actually tumors of germ cells, of stem cells, okay? And what happens is that these are slow-growing tumors, but because they start out as stem cells, the individual cells within that tumor can differentiate into all these different things. And so you can pop out this lump of tissue that's got teeth and hair. It's got, if you look under the microscope, it's got It's got respiratory epithelium, it's got nervous tissue, it's got, characteristics of the stomach and the brain and the eye all at the same time. All right, it's incredible, but this is what happens when stem cells go haywire. All right. They're also called dermoid cysts. All right, some facts about cancer. So different cancers are associated with different mortality rates. Just because a cancer is very prevalent doesn't mean that everybody dies from it. Skin cancer being an example. All right, it actually is the most prevalent form of cancer, but many forms of skin cancer are curable. Lung cancer is actually the leading cause of death in the United States. All right, cancer is Latin for crab, can be also called a malignancy or malignant tumor or neoplasm. Okay, causes of cancer. All right, so I really quickly apologize. Went through the machinery, the mechanisms that regulate cell growth. And so cancer itself is any alteration of that process that causes the cells to grow out of control. All right? And so it's a lot similar to a speeding car that can't stop. Okay? And you can have a car that's running out of control for a number of reasons. It could be that the accelerator is stuck. It could be that you have no brakes. It could be that you have both. All right? By the same reason, if you saw how complex the cell signaling pathway is, you can muck around with any step in that process and cause the cell to basically grow out of control. All right? Any genes whose disruption can trigger a tumor growth has been traditionally called an oncogene. Okay? And let me give you an example. This is called the RAS oncogene. And so remember I was talking about the tyrosine kinase receptors. All right. One of the first molecules that responds to an activated receptor is this protein which is attached to the inside of the cell membrane called RAS. And I forgot what RAS stood for. Do you remember? Okay. But RAS is a molecule which, when it comes close to an activated receptor, will bring together the rest of the machinery to cause signaling inside the cell to occur. All right. So that's normally what would happen, except if there's a mutation in the RAS gene. All right. Then the RAS molecule can sometimes be turned on permanently, regardless of whether the tyrosine kinase receptor is been activated or not. And so basically, by itself, it's kind of short-circuiting the process of signaling and just telling the cell, grow, 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 because it's mimicking the signal that would come from the epithelial growth factor receptor. Okay. Does that make sense? There's also something called Twohit Hypothesis, because if you remember, in your chromosomes, you have 2 copies of each chromosome. So that means that you have two copies of just about every gene, including RAS. Okay, so when I said that you can have a mutation in the RAS gene, you can have a mutation in one of the RAS genes, but the other one that you have in your body or in every cell is still okay, which means that you've only lost function in half of your RAS oncogenes. So you might be able to survive with one partially functioning gene and one not functioning gene. So in order to really have cancer, you'd have to knock out both copies of the RAS gene. Okay, so this is a hypothesis that had come out of early studies trying to characterize cancer. And so that theory of the Twohit hypothesis had been extended to kind of try to explain how cancer ultimately occurs. All right, because there's damage that occurs to the DNA in all of our cells all the time. Something like 10,000 points of damage to every chromosome in our body every day. Okay, and there's a really elaborate machinery that's involved with constantly checking the DNA to make sure that there's no damage. That if there are points of damage, it'll go and repair it. Okay, but sometimes those mechanisms get overwhelmed, and that's when cancer occurs. Okay. In terms of characterizing cancers usually characterized by the type and the stage. All right. You can have a cancer which is well differentiated or poorly differentiated. An example of a well differentiated tumor or a poorly differentiated tumor would have been, say, a tumor of like an early stem cell. All right, where you can't tell what it is. It could have been lung. It could be brain. It's really brain is growing out of proportion to the rest of the anatomy. In terms of stage, you could have something which is early stage or late stage, meaning that you caught it early in its development or late in its development. And so doctors have this system, they call it the TNM system of evaluating cancer, which gives you 3 pieces of information. Number 1, T stands for the size and extent of the primary mass. N is the number of lymph nodes that get involved, because typically what happens when a cancer spreads is it tends to spread through the lymphatic system in the body. And so if you look at the lymph nodes, that's normally where they will filter out cancer cells, and you're likely to spot spreading cancers at the lymph nodes. And then M is the extent of the metastases on a scale of 0 to 4. So what are the major tasks in cancer medicine? The major tasks, first of all, is identification and diagnosis. All right. If I give you a piece of tissue, all right, can you tell that it's cancer? Is it benign or malignant? What's the type or stage? Okay. Another job that you might be asked to do is to localize or to track the tumor. All right. Where is it located in the body? Where is it going? Okay. If you know all this, then probably your next responsibility is to treat it. And here, in treating cancer, the basic goal is to eliminate the cancer without harming the rest of the body. And finally, if all of this is mastered, maybe you'll know enough to prevent cancer from happening in the first place. Okay. And in all these cases, we think that biophotonics has a definite role in helping out. Okay. And this is an introductory slide you may have seen already, but why use biophotonics? It's because light is an extremely powerful tool in medicine. All right. In terms of carrying information, there's a lot of information that you can store in light that you can get from simply shining light on tissue. The fact that it can be used to perform very small and delicate tasks is shown here in this slide. That kind of shows you the scale of biology, going from single atoms all the way up to the whole body. And then down here is a number of different research techniques that we've had in the past to study biology. Okay, the problem is that there's been this gap that's right around the level of 250 nanometers and below, and this is right at the scale of molecular biology. So everything that we've told you about molecular biology, the problem is that we've never actually seen it. All right, we've inferred all these processes through doing analytical chemistry, through doing molecular biology techniques, but no one's actually ever seen DNA get replicated, like directly, right? And so what we're interested in doing is using biophotonics to actually produce a microscope that can see all these processes happening in real life, because nothing's never as persuasive as being able to directly see all these things happening, right? Seeing is believing. So how do we apply biophotonics in medicine and in cancer? Well, first of all, we can use biophotonics to understand the fundamental mechanisms of disease. All right, with high-resolution molecular imaging, the ability to work with living systems as opposed to, say, X-ray crystallography or electron microscopy, where you have to process the tissue and you've frozen it in time. So it's a snapshot, but you never really get to see how things are moving around and what's relating to what and how they're interacting. In terms of localizing a disease within a population or inside the body, biophotonics can be very useful because you've got advanced labeling methods, using fluorescent antibodies, which I'll talk about, as well as label-free methods, which don't require you to add any kind of fluorescent probe, just looking at the intrinsic optical properties. And then finally, developing more effective or efficient ways to treat disease. I'm going to show you a couple of techniques where we're using either fiber optics or something called photodynamic therapy to actually treat cancer. So let's hit diagnosis first. So conventionally, how do we diagnose cancer? Well, a doctor would have to do a physical exam and then look for certain signs and symptoms. Okay. So it could be that somebody feels a lump or they have a pain in their body. All right. There could be fever, fatigue, some unexplained weight gain or weight loss. All right. Or there could be some alteration in their metabolism. All right. Typically, if a doctor sees all this, they might request that they do some kind of imaging using X-rays or CAT scans or MRIs. And then there are certain features that you'll see on a picture, something that looks like bone, but there's no bone in the body that should be there, so it may be actually calcium that's deposited inside a tumor. Right? So there are certain signs that you can get by medical imaging that would suggest that something is a tumor. right? But the gold standard ultimately is to go in there, open the body up, get a piece of that tissue out, and analyze it under the microscope, all right? And so direct microscopic examination has been the key. But are there ways that we can improve on these methods?
Take a look at SmartProbe, a new device that could change the way breast cancer is detected. At Lawrence Livermore National Lab, researchers are designing and fabricating this diagnostic needle in conjunction with Bioluminate Corporation in San Carlos, California. At a press conference in January, researchers demonstrated this SmartProbe. In the time it takes to give a routine blood test, physicians will be able to tell their patients whether or not a suspected lump or tumor is cancerous.
But in very simple terms, SmartProbe is a device that will measure the optical properties and electrical properties of tissue. And we can use that to try to determine the tissue state and type. And what it's going to replace is a conventional core biopsy, which is this size, with a very, very small device that's this size.
Clinical trials of SmartProbe should begin this spring, and the device could be in the hands of physicians within a few years.
Right now in this country, about 1.2 million women per year have breast biopsies, and about 75 to 80% of those biopsies are benign. And so the work that we're doing here at Bioluminate at Lawrence Livermore is to try to lower that number of negative biopsies.
SmartProbe utilizes technology originally developed for the lab's nuclear weapons programs. Along with breast cancer detection, SmartProbe could someday be used to detect prostate, lung, colon, cervical, and brain cancers.
Okay, so that was a little news feature that was describing a technology that we were working on a couple of years ago. The rationale for developing that kind of technology was that in breast cancer in particular, a lot of women will detect lumps in their breast and get it examined, and then the doctor will say, well, I have to biopsy it. Okay, and in terms of a surgical biopsy involves actually doing surgery to cut out that lump and then getting it inspected. Now, this involves a great deal of physical and emotional trauma because it takes about a month to get the results. And what the doctor there was saying was that over 80% of these biopsies end up being negative. All right, so it's a lot of worrying over nothing. So it would be great if we could develop some kind of instrument that, I mean, sure, it's painful to have a needle stuck in the breast, I'm sure, but would you rather have that, or would you rather have somebody go in with an ice cream scoop and take out a lump of tissue? So I think most people would prefer to have it assessed inside the breast rather than to have a portion of the tissue removed. And so the goal here was to develop a probe that would look at both the optical properties as well as electrical properties of the tissue within that lump to see whether or not it was malignant or benign. Okay, it was a great idea, except that it's been what, 2-3 years since that news report came out, and we didn't get anywhere. And the reason for that is because diagnosing cancer is actually extremely difficult. All right, if I were to show you, these are 3 different microscopic views of tissue. This is cervical tissue, all right, and showed you the different sorts of pathologies that can happen in the tissue. I mean, you'd be hard pressed to figure out, okay, what's the difference between this kind of tissue, and this kind of tissue, all right, without some. And remember, these are actually stained, so you can see the nucleus inside them, right? If these were unstained tissue slices, they would look like wax paper or something extremely kind of bland and not kind of featureless. And so the problem that we had with the SmartProbe technology is that there were all these different intermediate conditions, all right, things that weren't necessarily disease, but it wasn't healthy either. And so how could the SmartProbe actually distinguish between all these different kind of gray levels between healthy and disease? All right, and this is actually a section of a portion of the cervix taken out to show you the tumor is actually down here, and the rest of the cervix is normal. So this is actually from a different presentation that I gave, but these are looking at different methods of biodetection. I'm going to rip through this really quickly, but basically direct staining and microscopy has always been the way that we characterize tissues and cells to see whether or not they're cancerous. advantage is that, number one, it's the method that we're most comfortable and familiar with, and so it's our laboratory gold standard. The disadvantage here is that it takes time to get these results, and it requires the expertise of the individual microscopist or pathologist to recognize the problem. There are more modern methods of biodetection. These include PCR and DNA fingerprinting, depending on whether or not you know what gene to look for. And also something called immunofluorescence, which I'll talk about really quickly. Immunofluorescence the general problem here is how do you detect disease inside the body just as the disease is getting started, okay? And it's a lot analogous to playing that game Find Waldo, where, at a carnival, Waldo might be dressed in some red and white stripes, but at a carnival, how do you spot him right away, okay? And this was a figure just to show you that, there are a lot of people in there, a lot of people who could look like Waldo, but they're not. And you could, even presumably, if you did staining, right, which is what you're doing with those tissues, you might paint Waldo purple, right? And so what are the odds of finding a purple person inside a carnival? Not likely, but still, in a picture where you've got all this color going on, it's still not easy to spot. So ideally, you want to make him glow in the dark, right? Because glowing in the dark is really easy to spot from a control tower. But the problem is, how do you get Waldo to glow in the dark, right? If you were to use some kind of glow in the dark paint, you'd have to grab Waldo, paint him first, and then throw him back out into the carnival, which is kind of like vacuuming the floor, and then you got that dust bunny that won't come up. You pick it up, you throw it back down, and then you vacuum it again, right? Instead of putting it in the wastebasket. And so here, in terms of trying to find cancer, right, it's the same thing, right? You can't first identify the cancer and label it, and then say, aha, I can use microscopy and see it, right? You need for that stain to automatically go to the cancer and to nothing else. So what are the tools that we have? Well, it turns out that fluorescent antibodies is the key. And so fluorescent antibodies uses both light and biology at the same time. What you're doing is you're using antibodies that are purified from the body. And these are antibodies that might recognize a cancerous cell or some other cell. And if you were to tag these to a fluorescent molecule, then basically these antibodies will automatically stick to the cancerous cell and not to other cells. And so this allows you to spot yes or no, are there cancerous cells in your slide. Right. I'm running out of time, so I'm going to skip through some of these other slides. I'm giving you copies of this presentation so you can look at it later. But basically, fluorescence microscopy has always been a traditional tool that we use to identify tumors. But you can't use certain fluorescent molecules inside the body, and we want to be able to develop tools that allow us to see cancer inside the body. All right. And so for this, we have techniques such as hyperspectral imaging and multimodal microscopy. So this would be a photograph of a tumor that, if you were to stain it, might look something like this. But without having to use staining, we can use what's called autofluorescence, or the fluorescent properties of the tissue itself, to be able to distinguish tumor tissue from healthy tissue. Right. At the tour of CBST, you saw this technology, the laser trap Raman spectrometer, that allows us to identify different types of blood cells, about whether or not these are tumors or not. And so these are an example of the spectra that we get. You might see subtle differences, but so long as the differences are consistent, then we can use that as a predictor of what's cancerous and what's not. So all of that was to talk about diagnosing cancer. Methods to localize and track it. Again, medical imaging is very important. You can image the whole body, and if you know what you're looking for, you can identify where the cancer is located inside the body. There are advantages and disadvantages to each method. Sensitivity and specificity are always an issue. So you might have something which is very sensitive. It's able to pick up cancer every time it exists, but the problem is that it can't tell you when you don't have cancer, because it's always saying, yes, there's cancer, yes, there's cancer. So it's too sensitive, right, not specific enough. Again, the gold standard ends up being direct inspection, and so in order to help that, we're using biophotonics to develop advanced microendoscopes. So these are optical fibers that probably are not much bigger than a millimeter or half a millimeter in diameter. And you can insert these in the body. You can perform extremely exquisite confocal microscopy inside to see what's going on without having to do open surgery. So this would be this would have definite advantages over the smart probe that we saw earlier. There's also techniques using bioluminescence. So this is using firefly luciferase. And so this process allows you to see tumors inside the animal without having to sacrifice the animal. So in terms of people developing drugs to see whether or not what's the effect of the drugs on the tumor tissue, right? You don't have to treat many animals and then sacrifice them at different time points to see kind of what's the progression of the tumor over time. But rather, you can just follow the growth of the tumor in just one animal. Okay. In terms of treatment, we have conventional treatments, which are surgery, radiation, chemotherapy. All right, there are advantages and disadvantages to all of them. Surgery is direct, but it's very drastic. You can't necessarily treat all metastases this way with surgery. Radiation is good in that it's non-invasive, but it ends up harming a lot of healthy tissue along the way. And again, you can't use it to treat all metastases. Chemotherapy is good in the sense that you can treat, metastases, but the problem is that not all tumors respond to chemotherapy. And then there's another newer technique called immunotherapy, which I'm not going to get into. In terms of biophotonics, what can we do? Well, there's a technique called photodynamic therapy, or PDT. All right, and so this is a technique where it's kind of a controlled chemotherapy. Suppose you have a tumor in your neck that you want to treat with chemotherapy, but if I inject you with chemotherapeutic drug, your hair falls out, your gums start to bleed, you get all these different side effects. And so one trick is to develop a drug which is inactive until it gets activated with light. And so I can take this inactive drug and inject it, and it won't have any effect on your body. And so it starts distributing all through your body, but only in the area of the tumor. If I were to shine a laser there, right, the laser will activate the chemotherapeutic agent, and so you'll have a chemotherapeutic effect only in the area of the tumor in your neck. So individually, the two components don't cause any harm to the body. The light itself doesn't harm your body. The inactive chemotherapeutic agent doesn't harm your body. But I put the two together, and it'll harm the tissue that's in your targeted area.
Okay, I think I just got this.
Okay.
Can you guys please give thanks to our speakers?
Thank you.